All right, ladies and gentlemen, welcome back. So we are going to wrap up this chapter by just talking sort of briefly about viral diseases, which personally I think this is kind of the cool stuff. So when we talk about viral diseases, they're really kind of interesting when we look across the way they interact with living things because they really do infect everything. Um, Most of what we study are either things that are just easy to access, so things like viral diseases that go after bacteria, because we can do it cleanly and easily and relatively safely in a lab, and then ones that tend to have big economic impacts. So ones that are affecting large portions of population, human populations, um, agricultural crops, livestock, that kind of thing. Um, the ones that are less popular in study and the ones we know less about are the ones that just don't seem to get as much funding because they aren't um, affecting something that is oftentimes monetarily important. Um, the other interesting thing is that we, up to this point, have talked about viruses in terms of infectious diseases being genetic information and protein. And yet there are these really interesting, sort of weird, sort of side pieces to infectious diseases. And I'm going to talk really briefly here about them, but they're kind of cool if you want to go spend some time digging into them. And those are these smaller, more um, minute things called viroids and prions. Um, and these guys still cause infectious diseases in plants and animals. And we always assumed that when we were talking infectious diseases, we were talking virus, bacteria, or some other parasite like a worm. And yet these guys have started becoming things that crop up here and there, and they will cause disease in plants and animals. So viroids are more often, at least as far as we know, plant pathogens, and they consist of just circular RNA. They go in, they disrupt a lot of their growth processes and things like that, but there's no protein code. So it's just genetic information going in, wreaking havoc, replicating itself, and moving on, um, which is sort of interesting, but in some respects kind of makes sense. It's got the set of instructions it needs in order to move. What maybe is more interesting are these things called prion diseases. And these are infectious agents that are just protein. So, how are they going and being able to replicate themselves and cause disruption um, when they don't have what we would traditionally assume is a set of instructions, like something DNA or RNA? Now, they do have a sequence of amino acids, so maybe that's how they're capable of replicating themselves. But what it really seems to be is an interesting just misfolding. Um, so we see these particularly in the brains of the organisms that they infect. And they seem to be very specific to mammals. So if you've ever heard of mad cow disease, that is one that affects cattle, and then it was cropping up in some humans as well. And that prion ends up being a misfolded protein that is capable of interacting with neighboring proteins and causing those neighboring proteins to misfold. And we don't fully understand what is happening other than it becomes this cascading effect of producing all of these misfolded proteins that sort of just glob onto each other and form these plaques, these kind of think of them almost like blood clots, but in your brain tissue instead. It's not blood, it's just this globbing of protein, misfolded protein. And if it's misfolded, that means it's not doing its job. And we typically find them in the cell membranes of neural tissue. And it's just very strange. But what it does is disrupts their ability to do their job, which means the brain function ceases. Um, and ultimately will result in the death of the animals that are infected. So this was a particularly concerning thing when it was showing up in livestock. And the question then became how much of it can be transferred to humans and what does it do in humans? And there's still a lot of questions surrounding things like mad cow disease, which is this prion disease. So just kind of an interesting field of research right now, looking at what that means. How does this work? Why does it work? What causes that initial protein to misfold in the first place? Because it is actually a protein that is naturally found in your body doing its job, and it just somehow spontaneously starts misfolding, and then that cascading effect happens to the rest of the proteins around it. So interesting, interesting disease. When we look at other animal diseases, we're talking about viruses that will often cause either damage to the cells or kill the cells. And most of the time, it's because they are releasing some sort of hydrolytic enzymes from their lysosomes. So go back to your cell structure, right? And lysosomes are things that your cells naturally make. They are little vesicles, little membrane-bound sacs full of digestive enzymes, hydrolytic enzymes. And your cells typically will merge those lysosomes with a food vacuole. So via that process of endocytosis, right, they'll bring in those big food particles. They need to get digested. They need to get broken down even further so the cell can actually use the components, either for metabolic processes like making energy or for other structural components. So the food vacuole will merge with the lysosome to break down that stuff, but it's all controlled within a membrane. Viruses will sometimes come in, and in part of their reproductive infection pathway, they'll cause those hydrolytic enzymes to get released from the lysosome itself into the cytoplasm, which is going to do all sorts of digesting of cell parts that the cell isn't looking to digest. Um, some other viruses can cause infected cells to produce toxins that then get released, and cause some of the symptoms we would associate with a particular disease. And others have molecular components, so proteins, carbohydrates, things like that, that are part of their envelope that are actually toxic. So that could be what causes those symptoms as well. So it's either directly killing the cell itself that it's currently within and infecting, or it can be producing other toxins that are getting released and causing damage elsewhere. So either it's localized damage, or it's systemic damage. So that first bullet there really is just looking at the localized damage. Of course, they're going to cause damage by just releasing massive amounts of them and exploding your cells too. So keep that in mind as well. The bottom two there are more the systemic issues that you'll see, further away from the initial infection site. You'll see symptoms in cells. So what do we do about it? Well, antiviral drugs exist. We're getting better at creating them and using them. Um, they'll help as a treatment, but they're not a cure. They're not a way for us to kind of eradicate, if you've ever heard that word, eradicate a virus, eradicate a disease. Um, So our best defense really are vaccines. And vaccines are harmless derivatives of that pathogenic microbe. And vaccines can be from against viral infections, they can be against bacterial infections. Um, but we create those vaccines either from genetic information, they can be component vaccines where it's just pieces of the antigenic proteins that are coming off of a virus or a bacteria, that your body can recognize and mount an immune response against. Sometimes there are killed vaccines, there are killed whole vaccines, so you'll get the entire pathogen. Um, and occasionally they're what we would talk about as attenuated vaccines, where we've severely weakened it, but it's still live. So you may get a very mild infection as a result of it. Though most people don't get symptoms at all. Regardless, they're safe. They are incredibly highly tested. Lots and lots of hoops to jump through, before vaccines are put on the market. Lots of testing, lots of people who have been tested, lots of organisms otherwise that have been tested. The big thing is the question of how effective is the vaccine, and some vaccines are better at protecting you than others. Um, and even from person to person, sometimes there is some variation in protection, but you will not get the disease from the virus, or from the vaccine, I should say, itself, um, other than maybe some very mild symptoms in those very few live attenuated vaccines that do exist. The point here is to get your body to learn that disease without ever actually getting severely ill from the disease first. So your body is capable of remembering things, at least on some level. And so our vaccines are a way to get you there. And keep in mind, I can't treat you with antibiotics. If you've got a viral infection, those antibiotics are specific against bacteria. So although we have some antiviral drugs, the vaccines really are the big thing. And they have to be given to a large enough portion of the population, too, to be effective as well, against ridding the population of the disease. So you as the individual may be protected by the vaccine, but if we are looking at large scale population level protection, there is often a pretty high percentage of the population that needs to be vaccinated in order for it to be effective. For a lot of vaccines, we're talking anywhere usually up in the 90+% of the population need to be vaccinated for it really to be helpful at a true population level. And then, of course, emerging viruses, emerging diseases. These are ones that sort of suddenly appear. They typically show up in small, isolated populations, but because we are such a global society where I can jump on a plane and literally travel the globe within 24 hours, it's becoming a much more global problem. So new viruses that emerge can easily get spread from one population to another, especially depending on how that virus transmits from one host to the next. Are you asymptomatic entirely, but you can still what we call shed the virus, mean pass on the virus? Are you shedding virus prior to showing symptoms, and so you don't even know you're sick and you've interacted with people and you've infected people? When we see viruses that gain a mutation that allows them to jump from an animal to a human, we talk about these as zoonotic pathogens. So something within that species, that host species, is changed just enough to now allow it to gain access to humans, which presumably those humans aren't dealing with that pathogen on the regular. They have no immunity, and now it's going to spread like wildfire. When these strains cause pandemics or epidemics, um, it's a big problem we've got to figure out how to deal with. Just to sort of give those two terms some definition, because they often, especially in the media, get used interchangeably. An epidemic is a local general outbreak of a disease, so within a town, within a state, within a country, but it's not global. Pandemics are when those epidemics have now popped up in multiple places across the globe. And so that's where that term pandemic comes from. These are just a couple of interesting books. This is not the end all be all. If you really get into this and really want some more recommendations, let me know because it's something I'm particularly interested in. But these are just 3 that I threw on here. So check them out. All right, last thing I'll leave you with, we have the tendency to spend way more time talking about viruses that occur in humans. And well, we're a little egocentric, if you will. We want to know what's going to affect us. That being said, viruses, again, infect all sorts of organisms. Pretty much everything that we know to exist has viruses that cause them disease. We tend to also pay attention a little bit more to plant viruses because a lot of the ones that we're paying attention to are ones that are showing up in our agricultural crops. So at this point, we know of more than 2,000 types of virus diseases that exist in plants, and most of those are the ones that are of some sort of economic interest to us, but they are known to cause spots on leaves, on the fruits, stunt their growth, damage their flowers, damage their roots, so they really can go after all sorts of parts of a plant as well. Most of the plant viruses have an RNA genome, um, so probably just a matter of the way that they've co-evolved over time. And they typically will spread in two major modes, either what we talk about as horizontal transmission, where they're able to get into one plant through damage in a cell wall, and then get to the next plant through the same means. So these are often plants that are grown close together or that have some sort of direct contact. Vertical transmission can also occur where a offspring can inherit the virus from a parent plant. We'll see a lot of that with organisms that maybe produce runners, or do some sort of asexual reproduction, maybe in addition to sexual reproduction if they do both. So unlike humans, it's usually one person to the next. This can also get passed parent to offspring. Uh, so it's just kind of an interesting additional mode of transmission, that we don't typically see with a lot of other viruses, and things like animals. Not that there isn't any vertical transmission that can happen, but it's not nearly as common.
Hi, it's Mr. Anderson, and this is the third podcast in the Life on Earth series. In this one, I'm going to talk about things that aren't really alive, and those are viruses. In 1982, Richard Skrenta, when he was a 15-year-old high school student, was sick of people taking his discs and borrowing his discs, and so he basically wrote a program called the ELK cloner that would copy itself onto the computer, and then it would copy itself onto discs that were injected into the computer. Basically, it would boot up the 50th time, it would give you this warning, this little poem, and it was really highly infectious. In fact, it was the first really infectious computer virus that was ever written. And so the program was able to move from computer to computer to computer. If there's no computer, then there's no computer virus. And so viruses work the same way. These right here are bacteriophages. And basically what they do is they inject their DNA into a bacteria. They use the machinery of the bacteria to make copies of themselves. In other words, they're going to make copies of the DNA. They're then going to take the DNA to make messenger RNA and then make proteins. And then those proteins are going to enclose the DNA, and then they're going to disperse, and they're going to move to another bacteria. And so viruses spread using cells. If there's no cell, just like if there's no computer, then there's no virus. And that's why we don't classify them as alive. They definitely have elements of living things, but they don't have metabolism. They don't have reproduction on their own without a cell to inject it in. And so basically, it's important to study viruses because they're one of the main sites of diseases that we have. In other words, millions of people were killed by the influenza outbreak on the heels of World War I. The 1918 influenza virus, this is a reproduction of what that virus looked like, or herpes virus, or polio virus, or look at all these viruses over here that infect us. And so basically, if it weren't for our cells, they couldn't they couldn't make copies of themselves. And so where did viruses come from? Well, we probably will never know the right answer to this, but here's a few theories. There's the cellular theory that they used to be part of cells, and so a plasmid, for example, is given off by bacteria, can be picked up by other bacteria, and so maybe they started that way, or maybe they were full-on cells that regress. So chlamydia, for example, is a type of cell that's lost the ability to live on its own, and so now it's an obligate parasite. Or it could have been coevolution, that these viruses were evolving with cells, just like insects and plants were evolving together. This is a little viroid, which is a little bit of RNA plant virus. And so there's evidence to suggest that any of these theories could have produce that first virus, but nonetheless, we eventually had viruses. So what are some characteristics in the structure of a virus? Well, all viruses are going to have nucleic acid, so they have to have some kind of genetic material. Sometimes that is DNA, and sometimes that is RNA. Sometimes that DNA is double-stranded, and sometimes it's single-stranded. And sometimes that RNA is normal, single-stranded, and sometimes it's double-stranded. And so these are all different types of genetic material that could be found inside a virus, but they're all genetic material. So that's the thing that all viruses have. The other thing that all viruses have is a protein coat. In other words, they have the coat that surrounds that virus. One of the first viruses really understood and crystallized and seen is the tobacco mosaic virus. And you can see here's the two parts of it. It's got RNA in a coiled pattern, and then it's going to have these protein subunits that go around the outside. So they protect that DNA. Now, a lot of the time, that whole protein is simply two different types of protein. We call those protein subunits. And you can see here that just having those two proteins, it can self-assemble into this shape, or this characteristic viral shape. And so all viruses are going to have these two things. But a lot of viruses are also going to have an envelope. So they're going to have a lipid bilayer. And so this right here would be human immunodeficiency virus, or the virus that causes AIDS. And you can see here that we've got RNA on the inside. We've got a capsid, or a protein coat, around the outside, but now we've got this lipid bilayer around the outside, or this envelope. Now, why is that important? If we say this is a typical cell, let's say this is a typical cell in your upper respiratory tract, and let's say you're being infected by a rhinovirus, or a virus that causes a common cold, well, around the outside of your cell is going to be an envelope. And so if we have another virus with a genetic material and a capsid, but let's say that they have an envelope around the outside, when it attaches to your cell, those two envelopes, if it looks exactly the same, is going to fuse with that, and it makes it easy for that virus to gain entry. Likewise, when it lyses the cell, or when it explodes, if it takes a little bit of that envelope with it, it's much more likely to be able to infect another cell. And so a lot of these envelopes are gathered from the cell in which they infected, in which they are leaving. So how do they reproduce? How does this actually work? Well, the viral life cycle is two parts. There's the lytic cycle. Lytic comes from the word lyse, or to break. And so let's kind of look right here at a bacteriophage. Bacteriophage, remember, infect bacteria. And so what this one is doing is injecting its genetic material, in this case DNA, into a bacteria. So it injected its DNA into a bacteria. As that cell makes copies of itself, you can see it's making copies of the DNA. Next thing it's doing, so how did it do that? It's using DNA polymerase. So it's using the machinery of the cell to make a copy of the DNA. What's going next? Well, right here you can see it's using that DNA to make proteins. And so how does it do that? Well, in this case, it's using RNA polymerase, and it's also using ribosomes. And so it's using the ribosomes inside the cell to make proteins. And so this right here would be transcription and translation. Eventually we have our genetic material, we have the proteins, and now we're going to lyse the cell. And so what's happening is these viruses are actually erupting from the cell. Where are they headed? They're headed to another cell to inject their DNA and make copies of itself. And so that's how you can just get a few viruses and you can get a huge viral outbreak, where pretty soon, exponentially, we're going to have billions and billions of viruses being created. And so that's one part of the life cycle. If you look at the second part of the life cycle, that's called the lysogenic cycle. In other words, it can generate the lytic cycle. What happens here? Well, basically they inject their DNA in, it becomes part of the genetic material of the cell, and then every time the cell makes a copy of itself, it's making a copy of that virus. And so that's kind of insidious. It's almost like a stealth mode where they're injecting their DNA. It's not actually making viruses, but it's using the machinery of the cell to copy it over and over and over again. So quick example in humans, because this is in bacteria. So if you've ever had chickenpox, that's a virus. It's infecting its DNA into the cells of your body. It's forming these pox, which is releasing more of the virus to infect more of the cells. And so it's going through this lytic cycle. Eventually, your immune system is going to shut it down, but this cell has injected its DNA and it'll actually sit in the DNA inside you. And so it can stay there, it can manifest itself decades later in another form of chickenpox called
shingles.
And so it's the same DNA that's just been hitchhiking around, waiting and waiting. So it's in that lysogenic cycle. And a lot of viruses will jump between the two. And so they'll be in the lysogenic cycle. Cold sores on the mouth would be an example of that. It's just hiding in your neural cells. All of a sudden your immune system gets depleted. They jump into the lytic cycle, make a bunch of copies of that, and then they can eventually go back. And so those are viruses. Are they alive? No. Are they important? Yes. And I hope that's helpful.